Hello and welcome to my first A2 biology video on nerves and the nervous system. So, the nervous system. Very simply, you will have a stimulus. Now this can be anything. This could be a change in light levels, you could hear a bang, you could stand on a pin. Receptor cells will pick this up. Now we'll come on to what sort of receptor cells you have later, but if it's light, you're going to pick up the light in your eyes through light receptors. Then it goes to the central nervous system, and it's usually the spine and the brain. And this is where all the stuff is processed. And to go from a receptor to the central nervous system, you have sensory neurons, and we'll come on to what they are later. Then the central nervous system goes to effector cells along motor neurons. Now sensory neurons connect to motor neurons via relay neurons. Effector cells are cells which will cause a response, so these could be muscle cells. So for example, if you want to pick something up, your muscles will contract to help you pick it up, and then a response happens. Sensory receptors. Now these are transducers. They convert energy into electrical energy, whether that be light energy from the eye, sound energy from in your ear, or pressure on your finger. It all converts into electrical energy which the body can use to transport the information. So the first neuron, sensory neuron. Now here you can see a number of features of a sensory neuron which you need to know. First you have a receptor cell. This is what picks it up. Then dendrites connect to a dendron. Dendron is what carries the electrical energy along a sensory neuron. Then you get to the cell body and an axon. Now the axon is always a lot shorter on a sensory neuron than the dendron. And then the axon connects to the axon terminals, which is the end of the axon, which goes to a synaptic knob, which is, as you'll see later, is the end of one receptor, and then there's a gap synapse, and then the new postsynaptic neuron. On some neurons, anyway, there are myelin sheaths. Now these are just layers of fat, which we'll look at later, which help speed up electrical conductivity. And as you can see, the direction of transmission is from receptor cells to the axon terminals. Motor neuron. Now this looks very different. You have effector cells. These are the cells that cause a response. Now this time the axon terminals connect to the effector cells, and you have the myelin sheath again, but this time you have a much longer axon. And basically dendrites take information, or dendrons are just big dendrites. Dendrons take electrical information to a cell body. The axons take it away from a cell body. You have the myelin sheath again, and also same as on a sensory neuron, in between two myelinated cells, which are called Schwann cells, you have a node of Ranvier, and this is where depolarization happens, and we'll cover that later. You have an axon hillock. That is where the axon connects to the cell body, and as I mentioned, dendrites. Now in this case, the direction of transmission is, it always goes from the dendrites along the axon, but this time it goes from the central nervous system to the effector cells. Now there's a few features that all neurons have in common. They are very long, so they can transmit the electron potential over a long distance. Some can be up to a meter in length. The cell surface membrane has gated ion channels that control entry and exit of sodium, potassium and calcium ions. This is particularly either at the synaptic knob or at the nodes of Ranvier. We'll go over what all this all means later. This is just the main structure. They have lots of ATP. They have Schwann cell. Not ATP, but ATP generators in the form of mitochondria, because they need ATP for active transport. They have Schwann cells. The cell body contains a nucleus, like always with many mitochondria. Motor neurons have their cell body in the central nervous system. Sensory neurons have it along their dendron near the central nervous system but not in it. And both sensory and motor neurons have numerous dendrites connected to other neurons. So, a nerve cell membrane. Neurons have specialized channel proteins for sodium, Na+ ions, and potassium, K+ ions. Now these can open and close. Now across a nerve cell membrane there will be a potential difference, a charge of about minus 60 millivolts, and this is called the resting potential, which we'll look at next. Now this occurs because of the transmission of sodium and potassium ions, and mainly occurs in the sodium potassium pumps. Now these use active transport because these go against the concentration gradient, and you have, for every 2 potassium ions which are moved into the cell, you have 3 sodium ions moved out. Now they're both positive, which means on the whole there's more positive on the outside of the cell than there is on the inside, so there's a potential difference across the membrane of minus 60 millivolts. This also occurs because inside there are negative ions as well. So the resting potential, this is when it's at rest. Now it's not actually at rest, it's not doing nothing. The ions are being transported across the membrane, as you can see. Now, you have the active transport of potassium and sodium ions in and out of the cell, but once the potassium ions have been diffused inside the cell, or actively transported inside the cell, they have a tendency to diffuse out where there are open potassium ion channels. At rest, there are sodium ion channels. These are closed, so sodium ions cannot return through. Now, an impulse is what transports the information. A nerve impulse is created by altering the permeability of the nerve cell to sodium ions. And what this does is that if you let the sodium ions diffuse back into the cell, then you will have a alteration of the PD, which will cause depolarization. Basically, the cell currently is polarized, it's got a charge. If you make the sodium ions diffuse back in, you change the charge and you depolarize it. And this will cause an action potential. How an action potential occurs. Now there are few things you need to know, a few definitions. What a voltage gated channel is. These are channels in the cell membrane that allow the passage of charged particles or ions, like sodium ion channels, but these will open and close in response to changes in potential difference. In particular, they normally open if the potential difference changes. The threshold potential, this is about minus 50 millivolts, so 10 millivolts more than the resting potential. If the depolarization of the membrane does not reach the threshold potential, then no action potential is created. It's kind of what is needed to make the action potential happen. If you do not get that high, it won't work. And then one actual potential is it's depolarization of the cell membrane, so the inside is more positive than the outside. Now this has a potential difference of plus 40 millivolts. It will always be, well, not exactly, but we say plus 40 millivolts. And what happens? Sodium ion channels open, depolarization, the voltage gated channels open. This allows a flood in of sodium ions, changing the charge, changing the PD. Then once it gets to a PD of 40 millivolts, the sodium channels close, potassium opens, and then repolarization occurs. And then something called hyperpolarization happens, and it goes back to resting state. We'll just very quickly explain that. Here is a graph, very roughly drawn. So you can see it goes up to 40 millivolts, it goes down again. Now it goes down further than the resting one of minus 60 millivolts. It might go down to minus 70, minus 80. This is hyperpolarization, it's gone too far. And what you can do is you can think of this as like a negative feedback system. What happens? A change occurs, so we have the potential difference increase. Then it decreases to alter the change, but it goes too far, so it needs to be changed again. It's often what happens. They overcompensate. Now, action potentials have something called all or nothing. This means that they will either have a depolarization which is large enough to reach the threshold potential, so you'll get the depolarization which reaches plus 40 millivolts, so an action potential will occur, or it will not. You won't have a kind of a half action potential. It won't just do some of it. The whole thing will go, or none of it will go. Now, as I said, sodium ions will diffuse into the cell, and you'll have potassium ions which will diffuse out. Now this means that they are in the wrong place. You've got potassium on the outside, sodium on the inside. So the sodium potassium ion gates will then work to actively transport 2 potassium in, 3 sodium out. And there's a small period of time after the action potential called the refractory period. This is when it's impossible to stimulate another action potential. It is just that, but it also makes sure that action potentials can only be transmitted in one direction, so you can't have it coming back. Now for the transmission of action potentials. Once you've generated an action potential, that's fine, but you need it to move across the whole body to the central nervous system. So how does this occur? Very simply, sodium ions can diffuse sideways. So the opening of sodium ion channels in one part of the neuron causes more sodium ion channels to be opened further down the neuron. So action potential occurs, the sodium ion channels open at one particular point, we'll call it point A. Then sodium ions diffuse across the membrane into the neuron. But as we said, this is the wrong balance of ions. This is a wrong charge. So the concentration of sodium ions inside the neuron rises. So sodium ion channels open, so the sodium ions can diffuse back out of the cell, but they will also diffuse sideways because there'll be a low concentration across the neuron as well, so they'll diffuse sideways, which then upsets the balance that area, so the ion channels open, sodium ions can diffuse, and it all happens again. Now, if you get the sodium ions moving across the neuron, there'll be less sodium ions inside the cell still than outside. So even though the gates have opened, diffusion will still occur into the cell, creating the depolarization. Now, some neurons have something called the myelin sheath. Now, these are made of fatty material, and they are electrical insulators. This means that the action potential won't just go all across into the cell and just die out. It'll stay in there. Now, sodium and potassium ions cannot diffuse through the fatty layer, so they have to diffuse at something called the node of Ranvier. Now, this is where depolarization occurs and creates an action potential. Now, this means that a neuron that is myelinated will do something called saltatory conduction. This means that the action potential will appear to jump from node to node. Now, the advantage of this means it is a lot quicker because it speeds up the transmission because it does jump, it goes quite quickly. So a myelinated neuron can conduct an action potential of up to 120 meters per second, which, considering the biggest neuron is 1 meter, it'll go very quickly. Now, a unmyelinated neuron is still very quick, but it's a bit slower, and when it's really needed, it can make all the difference. So, once a action potential has reached the end of a neuron, it reaches a synapse, a synaptic knob. How does it jump across? So this is what a synapse is. You've got calcium ion channels on it, mitochondria, smooth endoplasmic reticulum, vesicles consisting of ACH, which we'll explain, and then you have a postsynaptic. So this is a presynaptic neuron, and the one after the synapse is a postsynaptic neuron, and these have sodium ion channels, and between the gap in the synapse you have ACHE, and we'll explain what both of these are now. So an action potential arrives at a synapse. The voltage-gated calcium channels open and calcium ions diffuse into the synaptic knob. These then cause synaptic vesicles to fuse with the presynaptic membrane. These vesicles consist of a neurotransmitter. A neurotransmitter is a substance or a chemical that diffuses across the cleft of the synapse to transmit a signal to the postsynaptic neuron. And ACH, which is what we're interested in, these only occur in neurons which are called cholinergic synapses. And what this chemical is called acetylcholine, or acetylcholine, however it needs to be said. Now, the features of the synaptic knob, as I said, were mitochondria, because active transport is needed, so you need ATP. There's also lots of smooth endoplasmic reticulum for creating other substances. We have these vesicles of acetylcholine. Now, what these are needed to bond with the postsynaptic neurons' sodium ion channels. So the ACH is released and binds to sodium ion channel receptors. This causes the sodium ion channels to open. Sodium ions then diffuse into the postsynaptic membrane, and there'll be depolarization, and a new action potential will occur. Now, if sufficient generated potentials combine, then the potentials across the postsynaptic membrane reaches the threshold potential, and a new action potential is created. Now, this can happen from just one action potential, or it can happen from many, and we'll look at that soon. Now, you don't want the action potential to go on forever. You don't want infinite numbers of sodium ions going into the postsynaptic membrane. So you have an enzyme called acetylcholinesterase. This found in the synaptic cleft, and it hydrolyzes the acetylcholine to ethanoic acid and choline. Now, this stops the transmission of signals, so no more action potential can be produced. Now, this means that it stops the action potential, but also helps that move it in only one direction. Now, some few little kind of weird symbols to help you remember. Now, you can have two types of neurons. You can have converging neurons, which is when, let's say, 2, 3 or more neurons come to one other postsynaptic neuron. So many neurons release many neurotransmitters to one postsynaptic neuron. So you can see many to one. Divergence is the opposite, so you have one to many. One neuron releases neurotransmitters to many postsynaptic neurons. Now, something occurs called summation. Summation is a term that refers to the way that several small potential charges can combine to produce one larger charge and potential difference across a membrane. And there are two types of summation: spatial and temporal. Spatial is when you have, let's say, convergence occurring, and then these three small action potentials will combine all at once to produce one big action potential on the postsynaptic neuron. You could have temporal summation, which is when three small action potentials in very quick succession will arrive at one postsynaptic neuron, creating an action potential. So spatial, you can see they all kind of go at once. Temporal is one after another, each one getting bigger. Now, advantages of convergence is that if you're in danger, you have different areas of the body telling the central nervous system to do the same thing. So you could hear a noise and see a danger at the same time, and both will come and say you need to run, because they'll say which direction they are coming from. Divergence is very useful if you've got one signal that needs to go to many different parts. So if you have one response you need to do, but you need to do it in several parts of the body, divergence is useful. Synapses, they can also help ensure that signals travel in one direction, because you can't have sodium ions diffuse into the presynaptic neuron, and you also filter out unwanted low-level signals, because sometimes you'll get stimuli which you don't need to react to. For example, if there's a very small increase of temperature, you do not need to move your hand away. If there's a massive increase in temperature, you do, because you might burn yourself. And that is about it. So, this has been my first A2 biology video, so please say how you think I did on this one, because I've been teaching myself all of A2 so far. I've done Unit 4, Module 1 for biology and chemistry. So tell me what you think. If there's anything I missed out, just mention it in the comments, and say if you want more as usual. And I hope you're having a good summer and are enjoying doing this. So, conclusion. You have neurons, these transmit action potentials. You have sensory neurons, which go from receptor cells to the central nervous system, motor neurons, which go from the central nervous system to effector cells, and relay neurons, which go between sensory and motor. You have resting potentials, which is when when a neuron is at rest. It's about -60 millivolts. You have an action potential, which kind of when it gets excited, in plus 40 millivolts. Just to say, non-neuron cells can also have this charge. Transmission of neurons is caused by the movement of sodium ions along a neuron. Synapses help make sure that the action potentials are transmitted in one direction. And then synaptic transmission is caused by summation and convergence and divergence. So thank you for watching, and goodbye.